Hello, welcome to video 2 on photosynthesis specifically. This is going to be talking about just the light-dependent reactions. So let's go ahead and get started. It's actually not that many slides, but the material is so dense right now that it's very important that you spend time with this material, and at the end I'll give you some tips on how I think it's best to study. So just a reminder from what we already saw from the last video review, and when I talked about it in the very first lecture, there are two reactions that make up photosynthesis. The light reaction, which is what we're going to be talking about in this video, and the Calvin cycle. The light reaction is going to be the photo part of photosynthesis, and it produces ATP and NADPH. Now, if it produces ATP and NADPH, it's really convenient that ATP and NADPH are some of the requirements for the second part, and that's not an accident. So the light reactions are really important to proceeding through photosynthesis, because if we don't have ATP or NADPH, then we are not going to be able to proceed to make glucose. So we're going to talk about all of the reactions that we need to know for this course, with the understanding that there's a little more detail that would go in if you were taking maybe a biochemistry course. But it's very important that you're understanding the inputs and the outputs, and you're understanding where things are happening. If we don't start visualizing it, it's going to be very, very hard to memorize everything, because once again, this material is really dense. So I don't think the material is very hard, but it is not intuitive. And so it's very important that you're spending time with it to really learn it. Okay, so you heard me say ATP and NADPH. ATP you're pretty familiar with, but you probably don't know NADPH very well. And it's this relative to NADP+. They actually are going to be two sides of the same coin. NADP+ is going to accept electrons. NADPH is going to deliver them. So I want you to think of NADP+, NADPH as electron shuttles or electron taxis. Their whole job is to accept it when it needs to be accepted and deliver it when something else needs to be reduced. And so I want to remind you again of what reduction and oxidation is. Reduction is when something accepts electrons. Oxidation is when it delivers electrons. And so when we're thinking about this, NADP+ is going to accept electrons. So NADP+ is reduced. NADPH is going to drop those electrons off at some other molecule and then return back to NADP+. But NADPH, because it is delivering electrons, is going to be considered oxidized. So that is the whole job of NADP+. It's just a shuttle for electrons. Now, ATP is a little bit different. ATP is going to basically be a chemical currency. We are going to use ATP whenever we need energy to power a reaction. And the way that that works is, I want to remind you, well, ATP has 3 phosphates. It stands for adenosine phosphate. So 3 phosphates. Now remember that energy in biology is often stored in bonds. And so if I break this bond between the second and third phosphate, then I release energy. And since it releases energy, I can now couple this with a reaction that requires energy. So that could be transcription, that could be translation, that could be DNA replication, that could be photosynthesis. And so we break this bond between the second and the third phosphate. That's going to be releasing energy to be used for whatever we need it to be used for. But then that leaves us just with ADP, which is adenosine diphosphate, and this leftover phosphate. So we usually say P sub I. This stands for inorganic phosphate, and that's because this phosphate is not bonded to any carbons. So we have ADP now, and this is a lower energy molecule. So sometimes we want to generate ATP, or we get ATP back out, and we're going to see that in today's video. But when we turn it back into ATP, that is to store energy back into that bond between the second and the third phosphate. We're going to see a little bit more of this in cellular respiration, but I do want you to understand how ATP works as an energetic currency, because it releases that energy. We can spend that energy the way that we need to spend it for other endergonic reactions, because ATP hydrolysis is exergonic. Okay, so we've seen this image basically before. The light reactions are happening in the thylakoid membrane. They produce NADPH and ATP, which are going to be used by the Calvin cycle, which is happening in the stroma. Let's see how this works. So I asked you in class to write down these steps, but I thought it was more important that you're understanding what's happening when I was up at the board drawing it and up at the board explaining just based on the images. But the first thing that happens is light is going to be absorbed. So a photon is going to hit the photosystem 2, and then that is going to be transferred. The energy is going to be transferred to the reaction center. And then the electrons in the reaction center are going to be energized and move down the electron transport chain. While this is happening, water is splitting at photosystem 2. This is called photolysis. Then that water is going to donate the electrons to replenish the electrons that were there at the reaction center. Now let's see what I mean by this. First, we need to understand when I say proton, I really just mean a hydrogen that has lost its electron, because all that's left over is a proton. So you often will see H+, indicating a proton. You need to understand that this is a proton. If you don't understand that this is a proton, you will be very lost on some of these slides. But it's a proton because if we have a hydrogen atom which has an electron and a proton, and it loses its electron, all I have left is going to be a hydrogen ion, aka a proton. Okay, so let's go through those first two steps. First thing that happens, light is going to hit the light harvesting complex at photosystem 2. Now, this light harvesting complex is going to pass this energy from chlorophyll molecule to chlorophyll molecule, just like we see here with these yellow arrows. It is eventually going to pass it to P680, where there is an electron waiting to be excited. We'll talk about where that electron comes from in just a second. But the energy gets to P680 and it will excite this electron. Remember, I said excited electrons are high energy electrons that'll become important in the next couple of steps. But first step, light hits, passes the energy to the reaction center to excite the electron. Now, where did this electron come from? It came from water splitting. So water splits into half an oxygen and 2 protons, and what we don't see here, 2 electrons. Those electrons are going to go to the reaction center. That is where the electrons come from. Water is our source of electrons. Now you can also see that this is where oxygen is made. So one of the outputs of photosynthesis is oxygen, and it's coming from this step, so very early on in the reactions. Okay, now let's look at the next couple of steps. So we now have a high energy electron, and that electron is going to move down the electron transport chain. The electron transport chain is named very well. It is going to transport electrons from one protein to another protein. And this is very good because it is going to create a high concentration of protons in the lumen, but we have to understand how. Now, after it's transferred from one protein to the next, it'll finally, when I say it, I mean the electron, will be received at photosystem 1, which will re-energize it. So let's look at these two steps. I'll remind you, we just left off with a high energy electron because this electron was excited by the energy coming from light. So we have a high energy electron. Every time that it gets passed from one protein to the next, it is losing, and I'm putting that in air quotes, energy. It's not really losing energy, because what is happening is moving from a protein that doesn't really need the electron to a protein that really wants the electron. We say that these have high reduction potentials. And we say that the first protein maybe that's accepting the high energy electron would have a low reduction potential. Now, I know that those terms mean very little to some of you and mean a lot to others, but what I want you to understand, if we are moving from high energy electron and it's getting converted to a low energy electron, that energy doesn't disappear. That energy is going to be used to pump protons into the thylakoid lumen. So the thylakoid lumen is at the bottom here, and it is going to be filled by these protons that are on the outside. Now, you'll just have to believe me that there are sort of protons floating around everywhere, and so don't worry about where the proton comes from. They're kind of just everywhere in cells. And so as we move this electron down, we are using that energy from it being converted from a high energy electron to a low energy electron. We're using that energy to pump protons into the thylakoid lumen. Okay, now we have a low energy electron, and it will be received at the photosystem 1. Now, a low energy electron isn't accepted by very many things. It would have to be something that has a very, very high reduction potential. But we need to get that electron to NADP+. And so we're going to have to re-excite that electron. That is what's happening at 5. So the electron is going to be re-excited at step 4 at photosystem 1. Now we'll have the high energy electron again, which will be passed to NADP reductase to convert NADP+ to NADPH. So that's what's happening here. We have a high energy electron now because it got re-excited by a second photon, and we are going to be able to convert NADP+ to NADPH. Okay. Now what we're looking at here is basically the same thing, but it's going to be very important for when we're understanding how ATP is now made. So same thing we've already seen. Photosystem 2, we have an excited electron. That electron came from water, splitting water. That electron then is passed down an electron transport chain. It goes from a high energy electron to a low energy electron, and so that is going to be able to pump protons from the stroma into the thylakoid lumen. That low energy electron needs to get re-excited so that it can be passed to NADP+ to be converted into NADPH. Now we have a high concentration of protons in the thylakoid lumen, and that wasn't an accident. Now we can take advantage of this high concentration to make ATP. So how does this work? Well, it's actually a super cool mechanism. So, like I was saying, the lumen now is going to act as a reservoir for hydrogen ions, aka protons. Then the ions are going to move through an enzyme called ATP synthase. ATP synthase is going to turn, which will bond ADP to an inorganic phosphate to produce ATP. Let's see how this works. So, just like I was saying in class, just like a water wheel, the protons moving from the high concentration in the lumen to the low concentration on the outside is going to be exergonic, right? If we move from a high concentration to a low concentration, that is exergonic, that is spontaneous, that means it's going to release energy. So just like a water wheel, where it is physically turning the water wheel to power something like a house, it's the same thing here. The protons are actually physically turning the ATP synthase, and we are able to convert that mechanical energy from it physically turning into chemical energy stored in a bond that is in ATP. This is one of the coolest enzymes ever. It is going to show up again. I'm not done talking about it. I'm not done fangirling over it. It is an awesome enzyme. And if you take biochemistry, you will actually learn a lot about the structure in this. But it is awesome. Okay, so this process is called chemiosmosis. It is the idea of moving ions down an electrochemical gradient, specifically a chemical gradient. And so now we can see how we have successfully made ATP and NADPH from the light-dependent reaction. Okay, let's do everything all together now. So light from the sun in the form of a photon is going to hit the light harvesting complex of photosystem 2. Now it is going to pass the energy from one chlorophyll molecule to another chlorophyll molecule to another chlorophyll molecule to the reaction center to excite an electron that is waiting there. That electron is coming from water splitting into oxygen, protons, and electrons. So this electron will get excited. Now we have a high energy electron, and we are going to pass that high energy electron to a bunch of different protein complexes. And every single time it gets passed down, it is going to be using the energy that is, quote unquote, lost to pump protons into the thylakoid lumen. Now that will generate a high concentration of protons in the thylakoid lumen compared to a relatively low concentration of protons in the stroma. But now we have a low energy electron that we have to re-excite. And so light has to hit photosystem 1 to re-excite the electron to ultimately be passed to NADP+, to be converted to NADPH using an enzyme called NADP reductase. Now, taking advantage of this high concentration of protons in the lumen and low concentration in the stroma, we are able to allow protons to flow through ATP synthase just like it would in a water wheel to physically turn ATP synthase to form ATP from ADP plus an inorganic phosphate. This is the light-dependent reaction, and it is one of the coolest things in biology. Now, you also might hear this referred to as the Z scheme, and it's because you energize the electron, it goes down, and it goes up. So if you turn your head, it'll look like a Z. Okay, so I think this is a really cool animation. I'm going to let it play here for a second so that you can really see how awesome it is. But there's one more thing I need you to notice, and that is where things end up. Notice that NADPH ends up in the stroma. ATP also ends up in the stroma because those protons flowed back through to the stroma to form ATP. And that's exactly where the Calvin cycle is going to take place, the stroma. So it's not an accident that they end up there. I hope this makes everything a little bit more clear. Good luck in your studying, and I hope you have a great time. Ciao.